Hi everyone, my name is Sarah Taffel, and today we are doing another episode of UF DPM Programs Research Talks. Today we have Benjamin Waldo. And Benjamin, if you could start by introducing yourself.
Yeah, sure, Sarah. I am originally from Indiana. I grew up in a town called Bedford, and I attended the University of Evansville for my bachelor's, where I studied applied biology. Then I came down to UF, where I dual enrolled in DPM in a master's in hematology program, where I finished my master's in 2018, and I'm in the final semester of my DPM degree.
Well, congrats. As a DPM student, you're required to complete a thesis on your research over the years. Why did you choose to focus on this research?
When I came to UF, I wanted to dual enroll in a master's so I could get research experience. And fortunately, my advisor, Dr. Billy Crow in hematology, offered to take me on as his graduate student. So I decided to stay within hematology for my research, and I decided to investigate effects of nematicides on non-target invertebrates in Bermuda grass, which is a turf grass. And I was interested in ecology at the time, so I got to study a lot of different organisms, which was exciting for me because the main reason I came to the DPM program is because I'm interested in lots of different things. So I got to study nematodes, both the good ones and the bad ones, and then soil invertebrates like mites and springtails. So I got exposed to a lot of different things, which was really interesting to me.
And delving deeper into that, what were your methods in terms of collecting your data, and how long did it take?
So for this research project, we had turfgrass plots set up at one of the research centers affiliated with the University of Florida down in Citra, which is about 30 to 40-minute drive. And I helped apply nematicide treatments to these turfgrass plots. And then we evaluated over time changes in population densities and community structure. And to do that, we took soil samples at varying times after applying the nematicide. So we had some short term within like a couple days to a few weeks. And then we waited several months also before taking more samples so we could get kind of a timeline of how these invertebrates were being affected by the nematicides. After collecting the soil samples, we then extracted the nematodes and the arthropods, and we did that with a couple different methods, some being soil centrifugation for the nematodes, and then Berlesi funnels for the arthropods. And after doing that, I identified and counted them. And yeah, that was how we collected our data, and we compared over time changes in densities against for the different nematicide treatments to see if some were affecting these invertebrates differently than the others.
So what were the results in the end?
The results in the end were a little bit mixed. Some of the nematicides didn't really have much of an effect, which is good. That's what we were hoping, that some of these products were more targeted towards the bad nematodes. But some of them also affected the beneficial nematodes. And the way it works basically is with nematode studies, at least for studying beneficial nematodes, we look at different functional groups where we have like R strategists and K strategists, and that's based on largely what they feed upon. So some of the K strategists were more sensitive to some of the nematicide products than others. And we just, based on that information, we determined that some of these nematicides could be having negative impacts on the soil if you use them a lot over a long period of time.
So how can this research and these results help in real life?
That's a great question, because ultimately all research comes down to what does this mean in the big picture of things. And what this research is going to help with is that golf course superintendents that are going to be applying nematicide products now have a little bit more information on the possible ecological effects that these nematicides can have. So that can be useful for rotating or not overusing a certain product that could be having negative long-term effects in the soil, because if you wipe out some of these beneficial organisms, it's going to affect nutrient cycling, which then can ultimately affect the health of the plant. So hopefully over time, more of this kind of research can be done to support what we have found, or to help broaden the scope of the products tested or to see if they're finding similar results in other places.
That's a really good point. Thank you for taking the time to explain your research. And it's clear that there is a lot of time put into it and a lot of thought. So to finish up, do you have any advice to prospective DPM students on how to be successful when completing their own research?
Yes, research is definitely an interesting pursuit because you learn a lot of things the hard way. So I highly recommend DPM students getting involved in research, whether it's through a master's or a lab that they're working in, because there is no real substitute for getting your hands dirty and doing things on your own. And ultimately, that's part of what the DPM program is all about. You want to get your experience with firsthand work, and I would highly recommend trying to do something that pushes you and makes you feel a little uncomfortable, gets you out of your comfort zone. Becoming familiar with statistics is very important. It's not something that is necessarily always highlighted in the DPM program, but if you can get a firm foundation in statistics, in experimental design, that will help you for prospective jobs as well.
Thank you very much, Ben. I appreciate you zooming in today to share all this wonderful information to our viewers.
My pleasure, Sarah.
And thank you to all our viewers for tuning in for another episode. So stick around for more.